Welcome, Professor Stafaro here, and we are going to cover the next step in the cellular respiration process, the citric acid cycle. This cycle has a couple of other names that you might also come across. The Krebs Cycle, named for the man who won the Nobel Prize for discovering the cycle. It's also called the TCA or tricarboxylic acid cycle. So any one of those three names describes the same set of reactions. And that starts with grooming pyruvate as it enters into the mitochondria. So citric acid is our next step. Pyruvate that was formed in glycolysis will enter into the cycle, but first it must enter into a reaction that prepares the molecule to enter that citric acid cycle. So the first thing that happens is that a carboxylic group is removed and carbon dioxide is formed. Second, NAD plus gains an electron and is reduced to NADH. And third, a coenzyme related to vitamin B is joined to the 2-carbon fragment to form a molecule called acetyl-CoA. This is the molecule that can now enter into the citric acid cycle. So here's the cycle. This demonstrates a summary of the reactions, but let's just talk about some of the things in general first. The first is it's a cycle. As long as acetyl-CoA is available, the citric acid cycle will cycle through each of these reactions. Second, citric acid completes the breakdown of pyruvate, which essentially completes the breakdown of glucose. This diagram is broken down into several steps or parts, so let's take a look at the first step. The cycle begins when acetyl-CoA, a 2-carbon molecule, combines with oxaloacetate, a 4-carbon molecule, to form citrate, citric acid, the molecule the cycle is named for. Next, successive redox reactions lead to FAD plus and FADH to be reduced to NADH and FADH2. Again, these molecules will travel to the electron transport chain. Also, ATP is formed, and because we are breaking down the molecule, we also release carbons that are converted into CO2, carbon dioxide. Finally, the cycle regenerates oxaloacetate so that it's ready to accept another acetyl-CoA that's come into the mitochondria from the grooming of pyruvate. So in one cycle, hydrogens from acetyl-CoA and other intermediates are reduced to form NADH and FADH2. These two molecules are going to travel to the electron transport chain to help form ATP there. Carbon dioxide is formed and glucose is completely broken down. 1 ATP is formed in one cycle, and the cycle regenerates oxaloacetate and it's ready to accept another acetyl-CoA. Let's watch the animation on the citric acid cycle, and then we'll cover electron transport chain and oxidative phosphorylation. Okay, see you soon.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.